I'm Wally,
I'm Shannon.
and we're going to talk about endocytosis and exocytosis. So what's happening right now is movement from low concentration up to high concentration, and this is what active transport is, and that's what endocytosis and exocytosis is.
This is a lunch break.
Alright, so what just happened right now is one of the two first main forms of endocytosis, and it's phagocytosis. And what happens is that the particle, it's when the cell ingests large particles. So what's happening is that the cell here, it's getting near the cell membrane and it's ingested by the cell membrane, and the cell membrane becomes a little pocket, and then it closes back up and the little pocket becomes a vesicle. And that vesicle is then eaten apart by lysosomes, and the particle is then able to roam freely throughout the cell. And this is how unicellular organisms eat. So the second form of endocytosis is pinocytosis. And as you can see with this little cup of water right there, it's with fluids and solvents. So it's basically the same exact thing, just instead of large particles, it's with fluids. So the next type of movement with vesicles is exocytosis, and it's the reverse of endocytosis. So what happens is that the particle, instead of going through the cell membrane into the cell, it moves out of the cell through the cell membrane. And it's the same exact thing through the vesicle. It goes to the first layer, and then it just kind of leaves. And that's what exocytosis is. And cells use this when they need to get rid of waste products and toxins, etc. So now, going to our questions, Shannon, take over.
Question time! Okay, so what are the two main types of endocytosis? Think it over, discuss. Alright, question 2. What does active transport do? Discuss with your peers, think about it, long and hard. Alright, question 3. What is the difference between phagocytosis and pinocytosis? It's the last one, and we're outie.